So, as I said, this is a kit. It's called the P-Glow system. But what actually we're going to be doing today is bacterial transformations. Okay? So we're going to take bacteria and we're going to transform them, right, to do something that they don't normally have the ability to do. So, here's some examples of transformation using these fluorescent proteins that we're going to use today. And so this is an example of course, eukaryotes, mice, and they're shining the UV light on them, and you can see that two of them have the green glow even to their eyes and to their skin. And this is because when they were embryos or single-celled zygotes, they transform them by adding this additional DNA to produce this green fluorescent protein. And a lot of times they're doing this to tag them so they can tell if the process worked, right? Because you can't just look at an organism and know that their DNA has changed unless you give them something that is visual, that they can make from DNA that you could visualize. So in this case, green fluorescent protein. This is one with bacteria, right, and lots of different fluorescent proteins. And so they had a little bit of fun, right, and they actually drew a nice little tropical scene with different bacteria that are expressing different fluorescent genes. So again, what would be the purpose of doing this? That's kind of fun and pretty and artsy. This link, which hopefully will work for me this morning, hoping we can see it with the lights on. Yeah, okay. So you see those little green things moving, and then you can see also the red ones. They used two different fluorescent proteins for this experiment that they did. One of them green for the male fruit fly, one of the male fruit flies, they made his sperm green. And then the other male fruit fly, they made his sperm red. And then they mated with the female, and this is actually in the female's reproductive tract, the sperm moving, okay? So I'll play it again for you guys. And so they have several videos of this, but again, the reason why they made it colored so they could tell one from the other, because how could you tell one guy's sperm from another? You can't, right? In this case, they genetically altered it so that they were colored so they could see it. And they could visualize what was going on in the female reproductive tract. And they actually found that they compete. They compete to get to the egg. And of course, the female reproductive tract, they did further studies also, and has different things, chemicals in it to stop this competition. Because she wants her eggs to be fertilized by several different males, because then she has better chances of her offspring surviving, right? If they just mate with one male, right, if he doesn't have great genetics, then her babies aren't going to have great genetics, right? Where if she mates with multiple males, which they do, then their offspring are very diverse, and the chances of them surviving is greater. So very interesting stuff. So these types of fluorescent proteins have been used for genetic tags for a lot of different experiments if you do searches on the internet. I find this one kind of cool. When I talk about it with the junior high and high school students over the summer, they, the minute you say the word sperm, they pay attention to you. So, but they've used it in lots of different stuff. All right. So what is transformation? This is where you have DNA, and typically in the environment this happens. You'll have a bacteria that'll die. Its DNA will become part of the environment. It can be taken up by a living cell and incorporated into their DNA. And so what we're going to do today is we're going to put DNA in the test tube with the bacteria, and we're going to make the conditions such that the bacteria is going to want to take up that DNA and incorporate it. And how are we going to know if it actually did that? We're going to visibly be able to see this change. They're going to fluoresce green under UV light. And so we'll be able to see if that has happened, right? So a transformed cell has taken up a portion of DNA from another organism, right? In this case, it's going to be DNA that was originally in jellyfish. So in order for this to happen, the cells have to be willing to take up that, and we refer to that as competence, be willing to take up the DNA. About 1% of known bacteria are naturally competent, have the ability to take up DNA from the environment. It's a complex situation. There are several genes that regulate this. For instance, Bacillus subtilis has been studied in this, and there's 40 different genes that are involved in the ability to be competent, right? Ability to take up DNA from the environment. Why would an organism do this, right? It's evolved as a way of repairing their DNA. So if their DNA gets damaged, if they pick up DNA from the environment, it may be DNA from their own relatives close by, right? And that would help them repair. They don't have as many copies of their information as we do, so they're really susceptible to damage, and damage could be lethal for them. So this is one way of still surviving, picking up DNA and maybe repairing what you have lost. And this is often going to happen in late logarithmic growth. So this happens in an artificial culture system, what we refer to the different stages of growth. But it can happen in a natural system too. And it's when they are running out of nutrients, when conditions are harsh, they're more apt to pick up DNA from the environment under these types of conditions. And again, lots of cells, chances are they'll be able to pick up what they need from their friends nearby. Sometimes it can even be considered sexual reproduction. Not truly, right? But what sexual reproduction is an exchange of genetic information. In this case, they're picking up genetic information. And that information is passed to the next generation. In the laboratory, we're going to be using specialized competent cells today, but one of the ways that they can make cells competent is electroporation. So they're actually using an electronic field to create holes in the membranes of the cells so that they'll pick up whatever it is we want the cells to pick up, whether it's a drug or DNA if we're doing transformation. This can be used for prokaryotes as well as eukaryotes, those cell types. And so, of course, we don't utilize this in the lab because you need specialized equipment, right, with specialized cuvettes to put the organisms in and apply the electrical current. We use chemically treated cells. So these come pre-treated, and we're going to utilize a chemical solution. We're going to be using calcium chloride solution. This is the transformation solution. It's already in the tubes that you guys will be supplied today. We're just going to add the special strain of bacteria that are affected by this chemical, the E. coli, that we'll be using today. So we'll be using those calcium ions really help in the penetration of the DNA into these cells. We're going to do it by a chemical means. So the belief is that those positively charged calcium ions help with the negatively charged ions associated with the membranes and helps the DNA pass through. Only one strand of the DNA goes in. Okay, same thing for regular transformation. And then inside it'll copy the information and make it double-stranded. In this case today, right, as I said, we're using a specialized set of chemically competent cells. There are different kits that you can buy commercially available if you're working in a research laboratory. And there's different levels of what we refer to as efficiency, how good they are at taking up the DNA. So there's, of course, highly efficient ones, medium efficiently ones, and ones that are used for subcloning. This is where, Peter, remind me, subcloning, what are they doing?
Taking a portion of a gene.
Yeah.
Instead of a whole gene, just a portion.
Okay, just a portion of a gene. Okay, so if they're sub meaning just a part, right? So they're not looking at a whole gene, they're looking at pieces of genes. And commonly this is done when they're trying to do, what's the word?
Actually, I think sometimes the original clone may have multiple genes.
Yeah.
And from there, they whittle it down to the one gene there is.
They're looking for. But also they'll use it when they're doing,
Maybe
trying to,
one mutations sometimes. You want just to replace one amino acid.
if you want to know all the genetic information. So they'll,
No, sequence.
sequencing. I don't know why that would not come to my head. So like if you have an organism and they just chop up the DNA, they'll take pieces of it and put it into different subclones, and then they send it off to be sequenced by a lab. And so they're doing just little pieces of DNA, and then they'll get that information back and use a computer program to piece together the genetic information for the organism. Right, so it's one way they can sequence an entire organism. So I was working in a lab at LSU that they were trying to do this for a particular bacterial species. And it's been done for a lot of bacteria, especially ones that are studied in laboratory conditions. So we have little aliquots of the organism. They're stuck in the freezer and this special type of media. And then you take them out one at a time and they're ready to go. And then, of course, it's supplied with a positive control so that you're sure if it didn't take up DNA that it might necessarily not be the kit, just the pieces of decent DNA that you're working with, right? So we always have controls, right?
Today, we're introducing a different plasmid,
Right.
plasmid.
Yeah.
But researchers have found that pump plasmid is a great control, but it's always going to go in with your cells.
Yeah. So that particular control plasmid is a really good one. It usually always works. Always goes in. Always goes in. So bacterial transformation, what's the definition of this? This is the process by which bacterial cells uptake exogenous, so it's coming from the outside, DNA molecules. And they're usually not part of the organism's normal genome. It's genetic information. They have to be competent, and competent just means they have the ability to take up the DNA, right? The cells aren't always have this ability. They're competent. If they're competent, they'll have that ability to take up the DNA. And then one of the things that we're going to give them the ability to do today is related to bioluminescence. And this is the production or the emission of light by a living organism as the result of a chemical reaction in which the chemical energy, in this case, is converted to light energy. And so the fluorescent proteins that we utilize in research were originally taken from jellyfish. So that genetic information. And so sometimes if you go to the aquarium and you'll see the jellyfish are in kind of dark tanks, right? And some of them may be under UV light, so you can see these fluorescent abilities. I can tell you one of them that's really pretty. It isn't a fluorescent ability. It has little tiny like cilia on its surface, and because they're like little prisms, when they move, they refract the light, so it almost looks like they have like a moving rainbow that goes along them, right? And if you actually read the placard that's next to them, it tells you that, that's how they're able to do that. But some of them do have this ability, and that's where this gene that we're utilizing today originally came from. So how does this work, this fluorescent light? The blue light, the UV light that we're going to be using, is a high energy light, and it's absorbed by these organisms, and it excites electrons, and when those electrons, so they're going to be absorbed, they move up, and then when they move back, they fluoresce the green light, okay, which we can actually see. It's in the visible light spectrum, okay. You can't see UV light, right, but we can see the fluorescence, the retransmission of this light. And in the one that we have, it's in the green spectrum, which is really easy for us to detect. But as you can see, there are other ones that can be utilized. So this is just a chemical formula, right? Talking about how energy of light, right, can produce, wavelength of light can produce energy, right? So here is UV light, right? It's outside the visible light spectrum, but we're going to be able to see it because it's going to be retransmitted, right? That energy in the form of green light for our experiment today. So if all the conditions that I said this is a kit have been worked out for us, right? So we should always be successful. We know how many cells to use. We know how much DNA to use. This is all pre-measured out for us. We know the transformation efficiency, how efficient, how good it is at this process happening, right? How many transformants we'll get per milligram of DNA that we put into this kit. So, as I said, the volumes and everything are worked out for us. Normally, we'd have to make serial dilutions, right, and figure these things out. We don't need to do that for this experiment. This is the specially designed plasmid that we're working with, what's called the E.coli plasmid. And so it is a circular piece of DNA with genetic information on it. And it has several different genes on it. It has a beta-galactanase gene, abbreviated BLA here. And this is an enzyme it's going to produce that can destroy ampicillin, right? So we're going to give them ampicillin resistance. We've looked at this before. E. coli is sensitive to ampicillin, correct? It should normally be killed by ampicillin. And in this case, we're going to give it resistance so it will not be killed by ampicillin. So that's one of the ways we can select today for organisms that have the DNA, is that they will only be able to grow in the presence of ampicillin if they have the DNA and they express that information. Green fluorescent protein, the GFP, is the other gene we're giving them. As I said, this is a protein derived from jellyfish that allows them to absorb the UV light and then re-emit the green light, the green fluorescent light. ARAC, regulatory protein. So this is a protein that is going to be involved in the regulation. So this is a protein that's going to be produced, and it's going to help regulate this DNA, what is actually utilized by the organism. And it's always expressed, it's always being copied. It's always making these two proteins, the ARAC and the BLA, the beta-galactanase enzymes. It's not under regulation. Unlike GFP, it's under regulation. So how are things regulated? So for DNA, you have these operating systems within the DNA. Because think about this, you don't always need the information in your DNA to be expressed, okay? Especially for us, we're multicellular organisms. You have red blood cells and you have liver cells. What makes them different? It isn't actually their DNA. They have the same DNA. It's what DNA they're expressing, right? And this is based on the environment of your body that tells it, okay, you're going to be a red blood cell, you're going to be a liver cell, right? The same is true for microorganisms, right? In the environment that they're in, they'll express different parts of their DNA based on what they need. So sugar digestion is very common, and we're going to be using a sugar digestion pathway here for these particular cells. But for E. coli in general, they have the ability to digest, and we've seen this, lactose, right? But they don't always have lactose in the environment to eat. So they're not always making the enzymes or even the proteins in their membrane in order to take in that sugar, right? Most people, right, we digest it, we absorb it in our small intestines. By the time it gets to the E. coli in the large intestines, there's no lactose left. So there's no reason for them to have those enzymes readily available, right, to be able to digest lactose. But they do have the ability to sense it, and if lactose becomes available, then they'll make those enzymes to digest lactose. So this is a similar system today that we're going to be using, except it's just a different sugar. It's arabinose. And so how does the cell, right, know to make those enzymes, right, to utilize that portion of DNA? A lot of these are under a control mechanism. Operons are a sequence of genetic material that functions in a coordinated manner, consists of an operator, a promoter, and 2 or more structural genes. Right? So what's the operator? The operator is the segment of DNA to which the regulator or effector is going to bind. So things actually literally bind to the DNA in a specific location. In this case, it's called the operator. The promoter is where RNA polymerase is going to bind, if it can, right? A lot of times the promoter is blocked in this instance so that the RNA polymerase cannot bind. Or sometimes it's always available. It just depends. What is RNA polymerase? As the name implies, it makes RNA, right? Because we've got to go from DNA to RNA before we can make the proteins. The structural genes are the ones that actually do something. So in the case of sugar digestion, these are the enzymes that actually will digest the sugar. And the GFP gene, as I said, codes specifically for the green fluorescent protein. So arabinose is the sugar that we're going to be utilizing today. And normally, so here's the operator. This is the segment of the DNA. The regulator is bound to it, which is the ARAC, right? That protein that binds, it's the regulator. And so it's binding, and when it's binding, it's not allowing the promoter to be available for the RNA polymerase to bind here and copy these structural genes. Now, these structural genes are to make the enzymes to digest arabinose, right, to digest that sugar. So arabinose actually is the effector in this case. It binds to the operator, right? I mean, to the regulator that's bound to the operator, excuse me, right? And it will change its conformation so that the promoter becomes available and RNA polymerase can now bind. So here we have the RNA polymerase binding, and then it's going to copy this information, okay, and actually make those genes to be able to digest the arabinose. What they've done with the PGLO system is they've used this operator, right? So they're using the operator, they're using this regulator ARC, and we're using the arabinose. So when the arabinose binds, the RNA polymerase can bind to the promoter and it can copy whatever structural genes that are there. In this case, instead of making the enzymes to digest arabinose, we've tricked it and we've put GFP there, green fluorescent protein. So only when the sugar is present do we get this chain reaction, right, of it binding to the regulator and the promoter being available for RNA polymerase to bind, and we produce green fluorescent protein, right? So only under this condition of having this sugar is the cell actually going to make green fluorescent protein. Does that make sense? Okay, so.
I just wanted a quick comment. I usually pronounce ARAC as era for arabinose.
Okay, that's easier than trying to jumble up all the.
It's the third gene in the group.
Yeah.
In the arabinose.
In the arabinose group.
C is the third gene.
But that one's not under control. Okay. All right. So what's the question we're asking here today? So the question is green fluorescence, right? And you're only going to see that if the gene is expressed, not if they have the gene, right? It's if they actually express it which means that they copy into messenger RNA and actually make the protein. Okay, so green fluorescence it only happens if you have expression of that genetic information, right, of the gene. And the reverse is true too. You can, if you have gene expression, you should end up with green fluorescence, right? You should be able to go both ways. So, and again, another way you could say it is no green fluorescence protein, therefore no expression, right? Not that they don't have it, it's just that they're not expressing it, okay? We have to have expression. So, under what experimental conditions do we observe fluorescence? When are we going to actually see this? Right? When it's expressed. When it's actually expressed. So we don't observe any fluorescence when we know the green fluorescent protein is being repressed, right? When that sugar is not present, right? When we're not able to express it. Okay. So BLA, I mentioned this earlier, right? So this is genetic information to beta-galactanase. This is an enzyme that will break the lactam ring that's present in penicillin-like drugs like ampicillin, and it inactivates that enzyme. That antibiotic, right? So it's no longer effective. It becomes ineffective. So before transformation, do they have this gene? Does E. coli normally have this gene? No. When we transform them, we're giving it to them, right? So they're going to have it.
Can I interject a comment here? When we introduce the plasmid, it actually takes a lot of energy to reproduce that plasmid. And a lot of cells would normally spit it out. It wouldn't be worth their while to keep it. But because it has an antibiotic resistance gene, and you put it on a plate with that antibiotic, there's good reason to keep that plasmid. That's the selective pressure it allows that plasmid to stay.
But how do they really know that it's antibiotic? Do they have a way of sensing its antibiotic resistance?
Well, that gene is always expressed.
It's
So
constituent.
if the plasmid goes in, it's going to make the beta-lactamase enzyme. And if they're on media that has
right.
ampicillin, they sure as well better have that.
Right.
So
Okay.
it's that selective pressure leads the plasmid.
Green fluorescent protein, right, the GFP gene, do they have this before transformation? No. We're going to give it to them, right? So they don't have it, we're going to give it to them. I don't know why the animation's not working anymore. Okay. So that would be yes, then no. Maybe it's coming. Okay. So step 1. And this step has been done for you. Right? The tubes have been labeled plus and minus. They're already in your bins that you have on your bench top. And so the plus one we're going to add the DNA to, the minus one we're not. Why are we not going to add DNA to one of the tubes? That's our control, right? Our negative control in this case, right? They already have 250 microliters of the transformation solution, that calcium chloride solution in them. We're going to get some ice, little containers of ice. We're going to add some water to it. I have some specialized floating racks back by our water bath there that you're going to use to put your tubes on ice. Steps 4 and 5. So using a sterile loop, we have plastic sterile loops in your guys' bins. You're going to transfer some bacteria from a starter culture that I'm going to give you into each one of your tubes, right? Both tubes are going to need to have, and at this point there's no difference between the tubes, so you can use the same loop, right? No sense wasting loops. Then I'm going to come around and I'm going to show you, and I'm going to shine UV light on a vial that contains the plasmid. What is the plasmid? What type of molecule is it? What are we giving our cells? We're doing transformation today, so what are we giving them? DNA. We're giving them DNA. Should it fluoresce under the UV light? DNA? No. The protein fluoresces, right? Not the DNA. So as long as there's no protein contamination of my sample, should you see any glowing? No, right? So this, this isn't a David Copperfield or Chris Angel magic trick. This is real science today. Okay? So only the product, only the protein glows, not the DNA that codes for the information to do that. Okay, step 7, I'm going to add the plasmid, right? Because this is the expensive part of the experiment. So I'll give it to you guys, and you'll go ahead and mix it up and discard the loop. If we knock over this vial, it would be very expensive. So here is our animation. I'm sure will work for us this time. Remember that this plasmid contains these 2 genes, as well as the gene for making the regulator, the ARC-C. So before transformation, do they have BLA? No. Are they going to have it if they are truly transformed? Yes. GFP, do they have it before transformation? Do they have this ability to make green fluorescent protein? No. After transformation, should they have the ability? Do they have the genetic information? Yes. So step 8, we're going to leave the tubes in ice for 10 minutes. Okay? And then you're going to transfer them, and we're going to bring our whole tray back to the water bath back there. At step 9, it tells you to label your plates, but we've already labeled your plates for you, partly because some of the plates contain different ingredients, and it's just easier for me to label them ahead of time and keep them straight. As I said, we're going to bring our whole ice bath all the way to the back to our water bath. Our water bath is set at 42 degrees Celsius, which is around 107 degrees Fahrenheit. You're only going to leave it in there for 50 seconds, so do not walk away. All right, you're going to time it, and then you're going to take the floating rack right back out of the water bath, and you're going to place it back onto your ice for 2 minutes. Why are we doing this? What's the purpose of transferring both of these tubes from ice to hot water to ice again? It's called heat shock, right? And so you probably even done this on vacation, right? When you go from the swimming pool to the hot tub back to the swimming pool, right? It's quite shocking to the system. Some people think it's fun, right? But in this case, we're doing it on purpose. This is heat shock, right? Where they're going cold, hot, cold. This makes them more permeable under these situations and more apt to take up the DNA. Yeah. We're stressing them on purpose. So step 12, after 2 minutes, we're going to just remove the racks out of the ice bath and just place them on our bench top at room temperature. Then I'm going to give you a tube that contains LB. This is just a broth media, food for your bacteria. And you're going to transfer that into both of your tubes. So you're going to feed both your organisms, right? And so we'll look at the pipettes, and if you take the lids off your bins, go ahead and take your lids off your bins. Tape to the inside of your bin is one of those pipettes with the markings on it that let you know the different markings and what they stand for. This pipette you're obviously not going to use. I have it taped there, right, so that you can look and feel. The other pipettes you'll notice in your bin are wrapped because they're sterile. We do not want to touch these with our fingers, just the bulb, right? Otherwise they no longer will be sterile. So if you don't place the pipette into the liquid for your DNA positive tube, so don't stick it down into the liquid, just drop it in. You can use that same pipette to then add food to your minus tube, right? So we can conserve, right, and not waste resources. We can just use one pipette as long as you don't contaminate it. So here's our pipettes, right? And so they're 1 ml pipettes. but we're measuring in microliters, so 1 mil in microliters would be 1,000 microliters. And you'll notice that there's a raised line right at the top of your pipette. We've used these before, right, when we did the MRVP to draw up 1 mil. But today we're going to be working with much smaller volumes. So you'll notice that there is an indentation right here, and then another indentation right here in your pipette. That's 100 microliters, and that's actually where we're going to draw up that tiny little amount to put onto our plates. For feeding, you'll notice that there's a raised line right here on your pipettes. That's where you're drawing up to when we're adding the media to each one of these tubes. You're going to do that twice. You're going to draw up for your minus tube, right, and then you'll draw up again from the sample that I give you for your positive tube, right? So these are the two markings that are important to be able to see where they are, right? And we need our good pipetters, the ones that are got a good steady hand for doing the pipetting today. So why were the vials incubated for 10 minutes? So we're going to add that food and we're going to leave them for 10 minutes on our lab bench. What are they going to be doing in those 10 minutes? They're going to be eating. We gave them DNA too, right? What do they do with DNA? They're going to use it, right? They're going to make messenger RNA and they're going to make proteins. Which two proteins are not under regulation and they're going to make? And in fact, they need to make this one, otherwise they're going to die. They need to make the BLA, right? And they will also make the ARAC, right? That regulatory protein that's going to bind to the operator, and this is going to repress the GFP. So it's actually going to stop it from expressing it. But it is definitely going to express the ampicillin, the BLA gene, the ampicillin resistance, the beta-galactanase. And in fact, we need them to do this because the next step is going to be plating them onto media that contains ampicillin. So if they don't make it, what's going to happen? They're going to die. Right? They've got to make this stuff. So as I said, we're going to transfer 100 microliters of our sample from our DNA positive tube onto our DNA positive plates. And we actually have 2 plates. And again, it's that second indentation on your pipettes. Make sure that you're clear, right? You understand where these markings are and ask for help if you need it. Again, we can use the same pipette, okay? Because you're just taking from the sample and putting it onto 2 plates, right? So you can use the same pipette. You don't need a new pipette for each plate. For the plus tube, right? And then we're going to go to our minus tube. Then you do need to switch pipettes, right? You need a new pipette. And you're going to take a sample from your minus tube and put it on your 2 minus plates, right? There are 2 plates that we're going to put that sample on. And then you're going to use a sterile loop. And this is just to spread them out, right? Spread out that media, the bacteria on that plate. And the same holds true. You can use the same loop for your plus plates and a different loop for your minus plates, right? So you should only need to use 2 loops. Does that make sense to everybody? Okay. So do not tape the plates together like they tell you in the lab manual. Instead, write your table number on all 4 of your plates. Place them on the front table, right, next to the 37 incubator, and we'll put them in the incubator. So obviously transformation could occur only if the pico plasmid was introduced into the solution, right? So which of our plates is going to exhibit transformation? The DNA positive plates. And we're going to have 2 of them. We're going to have ones that contain ampicillin and one that contains ampicillin and the arabinose sugar. So why does and why will the one containing the arabinose sugar fluoresce and the one that does not contain it not fluoresce? Well, why is the sugar important? Because the sugar has got to bind to the regulator, right? and allow the promoter to become available for the RNA polymerase to actually copy that information. So it, in essence, turns on the operon, this operating system that's associated with just the green fluorescent protein gene. So we're actually only going to have one pretty glowing plate. So what information is provided by the LB plate with DNA, no DNA added to it? So what did we say? Why were we not adding DNA to our control? And in this case, this is just plain LB. It's just the food, right? It's just media, right? So what should happen? They should grow, right? There's nothing in this media that's going to stop them from growing. And this is our control to make sure that our bacteria can grow on this media and that it survived through this experiment, right? That we didn't kill it off. It's capable of growing. Now the next plate, of course, is LB ampicillin DNA negative. So again, they don't have the DNA, they don't have the ampicillin resistance gene. But this plate contains ampicillin, so what's going to happen to these bacteria? They're going to die, right? And this is our control to tell you what? That ampicillin can kill the E. coli we started with, right? It verifies. It can't grow in the presence of ampicillin. So we have 4 plates today, 4 little cute plates to work with today that I relabeled to match our ABCD here. Peter changed it up on me, but that's okay. So our first plate, right, notice DNA negative, and our second plate, DNA negative, right? This one is just the food, just the LB. This one is LB and ampicillin. So remember, this one we should have good growth. These are non-transformed cells. Right? This one we should have no growth, right? Non-transformed cells. Our other control to show that ampicillin kills. This plate, right? DNA positive contains ampicillin. Will we have growth? Yeah, but only of the cells that were transformed. And not necessarily all of them are going to get transformed, right? Only the ones that are transformed that actually make the ampicillin resistance gene will actually grow. Will they glow when we shine UV light on them? No. Because what do they need to glow? Arabinose. Does this plate contain arabinose? No, just ampicillin, right? So the next plate, our last plate, contains ampicillin and arabinose. So again, only transformants are going to grow because we're selecting with the ampicillin, right? But because there is the sugar, it should be able to bind and turn on that operon, and we should get fluorescence, right, on just this plate.
So the ampicillin controls which one's going to glow?
Yep. Which one's going to glow?
Glow.
Glow. The difference is. Which one's going to fluoresce, right? So,
Did you say ampicillin or arabinose? What did you say?
Ampicillin.
She said ampicillin.
Oh, okay. Arabinose. It has to have the sugar. It's got to have the sugar, arabinose. It's bad that it's 2 A's. Okay, so for these plates, right, there's going to be no fluorescence, which means no expression. But some of these, it'll actually also mean no gene, right? The first two, we didn't even give them the gene.
You can't express something you don't have.
Yeah, you can't express something you don't have. So definitely no here. We're here, right, it's just not expressing it. Here, we should get fluorescence, which means we are expressing the green fluorescent gene. Oops, and that's our last one. So